Welcome, everyone. Today, we are going to explore the fascinating microscopic world of the parathyroid gland. Imagine your body has a highly sensitive security system that constantly monitors the amount of calcium floating in your bloodstream. Just like a thermostat in your house turns on the heater when the room gets too cold, your body has tiny sensors that release a special chemical when your blood calcium drops too low. In this lecture, we will uncover exactly how these tiny glands manage to do such a massive job for your bones, muscles, and nerves, building our understanding from the ground up. Now that we know what our topic is, let us understand why this gland is so crucial. The parathyroid gland acts as a highly sensitive security system for your body. It constantly monitors the amount of calcium floating in your bloodstream. When calcium levels drop too low, this system immediately detects the change and releases a specific chemical to bring the levels back up. This continuous monitoring and adjusting is absolutely vital. Without it, your bones would lose their strength, your muscles could not contract properly, and your nervous system would fail to transmit signals. It is a tiny system with a massive responsibility. Understanding its importance leads us to ask, where exactly is this vital security system located? You might be surprised to learn that the parathyroid glands are incredibly small. They consist of 4 tiny pea-sized structures hiding right behind your butterfly-shaped thyroid gland in your neck. Because they are so small, they are easy to overlook, but do not let their size fool you. Even though they are tiny, they are densely packed with specialized cells that work around the clock to keep you alive. Their strategic location allows them to constantly sample the blood supply and react instantly to any changes in calcium levels. So, if we were to take a tiny slice of this gland and look at it under a powerful microscope, what would we see? We would see a bustling microscopic city, which is what we call histology. Histologically, the parathyroid gland is primarily composed of distinct types of epithelial cells. These cells do not just float around randomly, they are highly organized. They are arranged in dense cords or clusters, creating a structured environment. Furthermore, these clusters are surrounded by a rich network of tiny blood vessels called capillaries. This extensive blood supply is essential because it allows the cells to quickly secrete their hormones directly into the bloodstream. Looking closer at this microscopic city, we encounter its most important residents. The most abundant cells in the parathyroid gland are the chief cells, which are also known as principal cells. Under a microscope, these appear as small, dark staining cells. They are the true workhorses of the gland. Their primary responsibility is synthesizing and secreting parathyroid hormone, commonly abbreviated as PTH. Whenever the gland senses a drop in blood calcium, it is these chief cells that manufacture and pump out the hormone to correct the imbalance. They are the active engines driving the gland's entire physiological function. Alongside the hard-working chief cells, we find a second, very different type of cell. These are the oxyphil cells. If you look at a histological slide, oxyphil cells stand out immediately because they are much larger than chief cells and they stain a bright pink color. This distinct pink appearance is due to an incredible abundance of mitochondria packed inside them. They tend to appear in clusters throughout the gland. Interestingly, despite their large size and high energy capacity, their exact function remains somewhat of a scientific mystery. We know they are there, but we do not yet fully understand their role in calcium regulation. As we complete our tour of the gland's cells, We must look at how the gland changes over time. As a person ages, the functional tissue of the parathyroid gland is gradually infiltrated by adipocytes, which are simply fat cells. These cells act as filler tissue. By the time a person reaches adulthood, these fat cells can make up to 50% of the gland's total volume. This means that half of the gland is essentially inactive space. This unique cellular composition, combining active chief cells, mysterious oxyphil cells, and inactive adipocytes, forms the complete histological profile of the parathyroid gland. Now that we have discussed the three main cell types, let us visualize them together in this diagram. What you are looking at is a microscopic cross-section of the parathyroid gland. Notice the dense clusters of small, dark purple cells. These are the chief cells, the tireless workers producing the hormone. Next to them, you can clearly see the larger, bright pink oxyphil cells, packed with mitochondria. The large, clear, empty-looking circles scattered throughout are the adipocytes, or fat cells, taking up space. Finally, weaving through all these cells, you can see the thin red lines of the capillary network, ready to carry the secreted hormone away into the body. This visual perfectly summarizes the gland's architecture. Having seen the healthy structure, we must ask what happens when this histology goes wrong. In the real world, doctors look at these specific cells to diagnose severe metabolic diseases. For instance, a patient might develop a parathyroid adenoma. This is a benign tumor that causes the chief cells to multiply out of control. When these chief cells overproliferate, they pump out dangerous, excessive amounts of parathyroid hormone. This leads to a condition called hyperparathyroidism. The excess hormone aggressively strips calcium away from the bones, making them brittle and highly prone to fractures, while the excess calcium in the blood causes painful kidney stones. Conversely, what happens if we lose these cells entirely? Because the parathyroid glands are so tiny and located right behind the thyroid, a surgeon might accidentally remove them during thyroid surgery. If this happens, the patient instantly loses their chief cells. Without chief cells, there is no parathyroid hormone being produced. This leads to a sudden and potentially fatal drop in blood calcium levels. Without enough calcium, the nervous system misfires, causing severe muscle spasms and even paralysis. Therefore, knowing exactly what these cells look like and preserving them is quite literally a matter of life and death in clinical medicine. To truly understand how this cellular composition affects the body, we can express the hormone production mathematically. This formula calculates the total parathyroid hormone secretion rate. Let us break down the symbols. SPTH represents the total secretion rate of the hormone, measured in picograms per minute. MT stands for the total physical mass of the gland in milligrams. The variable FA represents the fraction of the gland taken up by fat cells, expressed as a decimal. Finally, RC is the average secretion rate per milligram of active chief cells. Conceptually, this formula tells us that the total hormone output depends on how big the gland is, minus the dead space taken up by fat, multiplied by how hard the remaining chief cells are working. If the fat fraction increases, the total secretion decreases unless the cells work harder. Now that we have our formula, let us see it in action with a healthy 40-year-old adult. We are given that the total mass of their gland is 120 milligrams. Histology shows fat makes up 30%, so the fat fraction is 0.30. The healthy secretion rate is 50 picograms per milligram per minute. We need to find the total secretion rate. First, we calculate the active mass by subtracting the fat fraction from 1. giving us 70% active tissue. Multiplying 120 by 0.70 gives us exactly 84 milligrams of active chief cells. Next, we multiply this active mass of 84 by the cellular secretion rate of 50. This yields a final answer of 4,200 picograms per minute. Physically, this represents a perfectly normal baseline hormone production to keep blood calcium stable. Let us contrast that healthy example with a patient suffering from a parathyroid adenoma. Because of the tumor, the total mass has swollen to 500 milligrams. The rapidly dividing chief cells have pushed out the fat, reducing the fat fraction to just 5%, or 0.05. The cellular secretion rate remains at 50. We apply the same steps. First, we find the active mass. We multiply the mass of 500 milligram gland by 0.95, since 95% is now pure chief cells. This gives us 475 milligrams of active tissue. We then multiply this by 50. The final calculation reveals a staggering secretion rate of 23,750 picograms per minute. This massive flood of hormone will aggressively dissolve the patient's bones, demonstrating exactly why understanding histology is essential. To wrap up our journey into the parathyroid gland, let us review the most important ideas. First, remember that the small, dark chief cells are the tireless workers that produce parathyroid hormone to keep your calcium levels perfectly balanced. Second, the larger pink oxyphil cells are packed with energy-producing mitochondria, though their exact purpose remains a puzzle. Third, the clear adipocytes act as filler tissue that naturally increases as you age, changing the gland's physical structure. Finally, as we saw with our calculations, the microscopic histological composition directly dictates the massive physiological output of the gland. By mastering this histology, you now hold the key to understanding how tiny cellular changes create massive ripples throughout the entire human body. Great job today!